to grow without any pot. They're going to grow continuously. However, this we call somatic cell. But stem cell, okay, when you say stem cell, the stem cell must have these two features, okay. One is they able to self-renew, and second thing is they able to differentiate into a specialized mature cell. I will explain to you later why they have to have these two features. Okay, remember, in your body, you have a stem cells, right? Let's, for example, for bone marrow. In the bone marrow, you have a multiple type of stem cell, and one of them is hematopoietic stem cell. Remember, we don't have too many stem cells. We have in our entire body, in each organ, we have a limited number, a limited pool of stem cell. But let's say our blood, right? Our blood will be replenished every 3 months, right? 3 or 4 months. Every 3 months, right? I think every 4 months. Every 4 months, the blood of our body, the RBC will be diminished, and we will have a new blood, new RBC, right? So how the body will produce a new red blood cell every 4 months with a very limited number of the stem cell? Because in order to stem cell to become a mature cell to produce the red blood cell, that stem cell, or we can specifically say it as a hematopoietic stem cell, they have to propagate into a certain number. Okay, so they have to divide into a certain number. So some portion remain as a stem cell and some portion will differentiate into a RBC. So with this cell renewal means a process that make an exact copy of the stem cell. So before stem cell differentiate into a mature cell, the stem cell has to self-renew to produce and sufficient number of stem cell before to be used to mature into a mature cell. That's why cell renewal is very important. If you don't have a cell renewal, all the stem cell in your body will be exhausted. At the end of the day, you don't have any stem cell, then you die because of degenerative disease. Okay, that's why cell renewal is very important. And stem cell can be retrieved from many organs and many sources. The first one is embryo, and we also can produce stem cell from fetal tissue. Those fetal tissue which you can obtain from a clinically aborted fetus, we can produce a stem cell. And most importantly, we also can produce a stem cell from perinatal tissue. Peri means during, natal means delivery. So during the delivery, what you get is either you will get your placenta, umbilical cord blood, or umbilical cord tissue. So these three things can also produce a stem cell. And then once the baby is born, okay, that's we call post, okay, postnatal life, whereby every organ in the body will have the stem cell. And our talk will be more focused on adult stem cell. And adult stem cells are multi, oligo, or unipotent stem cell. I will teach you later what is a multi and oligopotence. And the common adult stem cell are actually come from the bone marrow, which is hematopoietic stem cell that produce blood, and also mesenchymal stem cell that involve in providing a stromal support to the tissue and also immune modulation. So in the brain, we have the neural stem cell. In the liver, we have an oval stem cell. In the muscle, we have a satellite stem cell. So basically, every organ in our body has a specific stem cell or tissue resident stem cell that take care of that tear and wear of the organs. Okay. And now let's talk about how we can classify. So we know that stem cell can be retrieved from the embryo, fetus, and also cord blood, and also from the adult. So when we receive the stem cell from different kind of a life phase, they have a different character as well. And one of the characterization of the stem cell is the ability to differentiate. Not all of them. Like your mother, right? Your mother, she's being mother to you. she's being a wife to your father, she's being a daughter to your grandfather, she's being a sister to her sister, a brother. So one single mother, but she has a many multiple facets, right? Same thing as well, okay? So a stem cell may have a different potency. This potency we call totipotent, pluripotent, multipotent, and progenitor cells. Like a zygote, the first cell that derived from fertilization of the sperm and ovum, we call zygote. Zygote as a totipotent. Totipotent means you just split the word toti and potent. Potent means potency. Toti means total. It means when you take the zygote and when you implant into a uterus, that zygote can develop into an entire human being. That's we call totipotent stem cell, okay, or zygote. The next one is when the zygote develop into a blastocyst. In the blastocyst, you have embryonic stem cell. This embryonic stem cell called as a pluripotent. It means they can become any cells in the body. However, when you implant this stem cell into a uterus, they cannot produce human being because they cannot produce the placental cell. They cannot produce extra embryonic layer, which is a placenta. Okay, the example of the totipotent stem cell are embryonic stem cell and also inducible pluripotent stem cell. Okay, these two are not so much important for us for today's talk, but what is important to us is a multipotent stem cell where you can get them from the fetal cell, perinatal stem cell, and postnatal stem cell. Okay, these are hematopoietic stem cell, mesenchymal stem cell, and any stem cell from the organ. So they can become a few type of the mature cell. It means like a pluripotent stem cell, embryonic stem cell, they can become brain cell, they can become a blood cell, they can become a muscle cell, they can become any cell of the body. But multipotent stem cell, they only have limitation for the differentiation. They only can differentiate along to their lineage. If the hematopoietic stem cell only can differentiate into a blood cell, like a red cell, white cell, and platelet. Like a mesenchymal stem cell, it only can differentiate into a mesodermal origin, which is a fat, chondrocyte, and osteocyte. Okay, so they have the limitations. Okay, like a cardiac stem cell, they only can produce a cell that in the heart tissue. Okay, and then we have progenitors. Progenitors are not a stem cell. These are and transiently existing cell in the system. So before stem cell become into a mature cell, the phase between the transformation, the translation of the stem cell into a mature cell involve the progenitor. So they basically will have a very limited differentiation ability. So I hope all of you understand what is that potency of a stem cell. So you should know that stem cell means it has two characteristics. One is they able to self-renew. The second one is they able to differentiate into a multiple mature cell type. And stem cell also have a potency based on where they derive from. It will have its own potency. If it is from that fertilization zygote, it is a totipotent embryonic stem cell, and inducible pluripotent stem cell have pluripotency, and most of the adult stem cell or postnatal stem cell, they have multipotency. Now, why stem cells suddenly become very what you call sexy, become very lucrative? Because two things happen in the entire world that change the perspective on the stem cell. The first one is Dolly. You know, Dolly is the first animal that been cloned. So the Dolly was born on 5th July 1996. It has three mothers. It means the Ian Milmuth and his colleague, they produced Dolly. So it's the first what you call cloning in animals, showing that three mother. Okay, it means three mother being used to produce a single child. It means ovum of one sheep are taken, and nucleus from the ovum of the another sheep are taken. These both are combined to make the zygote, and this has been implanted into a third sheep. So this is what we call cloning. It means that productions of the organism does not involve the involvements of the sperm and the ovum. It is just involved in a one somatic cell. It just involve the only single cell. Okay, that is we call cloning. And later in 2012, it's a very big boom, because usually what we know is when stem cell become mature cell, it's a one-way process. You cannot reverse the mature cell into a stem cell. It's always one-way process. And because of one-way process, we have a difficulty to acquire a stem cell for the treatment and research. But this wonderful man, Shinya Yamanaka from Kyoto, Japan, he shows that by using one somatic cell, the fibroblast, okay, he used the fibroblast, which is the most common somatic cell. By adding 4 different oncogenes, he convert the somatic fibroblast into a stem cell that has pluripotency. It means this pluripotent stem cell now can differentiate into any kind of a cell. So it gives a huge perspective that in future we can produce our organs or cell just can converting one of our fibroblasts into a IPS. It's all just a concept in the science and research. And this is just a simple explanation how embryonic stem cell being generated. So we have that fertilized egg with the sperm. It become a totipotent stem cell, okay, and it become an embryo. In the embryo, we have an inner cell mass. This inner cell mass, if you leave them, it become a fetus and human being. But if you take the inner cell mass, okay, if you disturb this embryo, you take the inner cell mass, and this become an embryonic stem cell. So this embryonic stem cell, okay, when cultured on the stromal cell or on the proper culture media, they can become any cell what you want. It can become a renal cell, it can become a liver cell or heart cell, whatsoever cell. Okay, and this is what we call the Sinmaya's work, showing that he produced that first what we call pluripotent stem cell from mouse embryonic and fibroblast culture. Okay, so what he has done is, okay, this is what we call inducible pluripotent stem cell or iPSC. Okay, why is this is so much of attract attention is it changed the concept of the stem cell. So basically what we know is we can produce the stem cell will be what we call harvested from that in a body, and then it become a mature cell. So the process is a one way. It cannot, there is no reverse, there is no two way. But he, with an excellent and elegant experiment, okay, what he has shown is he had taken that adult fibroblast cell or fibroblast cells, and then he add these 4 oncogenes, okay. When you add this oncogene and he reprogrammed the fibroblast and it become an iPS cell, it means the normal cell, the somatic cell, acquire a feature of the embryonic stem cell. So this stem cell and now termed as inducible because you induce a pluripotency by adding the 4 oncogenes into the fibroblast. And then this iPSC can be differentiated into any type of the cell, okay, by adding an appropriate induction media. Okay, so imagine that, okay, although this is not fully being translated into clinical, just imagine that you can take your fibroblast through your oral cavity, you add this 4 gene and bump, it become a stem cell. So let's say if you're having a kidney failure, okay, you don't have a kidney. So either you will get the kidney from the donor, now you have a chance to create your own kidney from your own somatic cell that been transformed into a stem cell. You just imagine, although this still like science fiction, many research are actually on the way to translate this into a clinical trials. And also, why iPSC is very important? Because iPSC can be also used as a disease modeling. like most of the time we have some what you call some rare genetic diseases or other genetic diseases where we can produce a specific patient specific iPSC, and we differentiate them into a specific disease cell, and we can use our target's drug to do a drug modeling, and also we can use this to the treating the patient. And same time, let's say a patient having a degenerative diseases, you can edit. Okay, nowadays now we have a lot of genome editing tools like CRISPR, and we have a lot of genetically modified or correcting method. So with that method you can produce the iPSC, and then you can produce a particular cell that can be used to as a cell replacement therapy. So we can use iPSC for two circumstances. One, to do a drug developments, to a customized drug development to a particular patient. So nowadays it's a precise medicine whereby the medicine, the treatment are given to you based on your own cell. Okay, not like generalized like you use Panadol for everyone, but this is very specific, a tailored kind of the what you call treatment that given to you. Or we also can produce spare organs or spare tissues to replace the organ that damaged or the tissue that been injured or damaged. That's the beauty of iPSC. So I didn't go so detailed on this part because it just a theory, and it also shows that the prospect and the potential of the iPSC in the modern medicine. And also like in the cell-based therapy, like skin fibroblasts can be reprogrammed into iPSC, and that can be differentiated into thyroid follicles, and that can be used for cell therapy, disease modeling, or drug discovery. So you do not need to use a human or sometimes an animal for your research. So you can produce a specific cell that cater to the patient to have a precise result for precision medicine. And now, so we have a lot of ideas. So we have a lot of discovery that we have iPSC, we have embryonic stem cell, and we also have adult stem cell. Okay, and then how this being translated. Okay, the translation has been done after conducting many preclinical animal study and in vitro study, and then it has been translated into clinical trials. Okay, clinical trial has 4 phases. Okay, before a treatment become a treatment, it has to go through many phases of the clinical trials, at least 4 phases. Okay, so after the stem cell research being undergoing clinical therapy, it can become into a legalized therapy. Okay, now, so I have done the basic part of the stem cell. So I have explained to you what is a stem cell, how do you define stem cell, and also the prospect of the stem cell in the modern medicine by giving an example of iPSC. Okay, iPSC is still a bit controversy. Why? Because the embryonic stem cell and also iPSC, when implanted into an animal model, it produced teratoma, a cancer. Okay, due to safety issue, the usage of the embryonic stem cell and also iPSC is very limited, and they are still under that research. However, there is another category of stem cell which I mentioned earlier, which called as adult stem cell. They don't have this teratoma formation. They are adult stem cell, and their differentiation are very limited. They are multipotential. Because they're multipotential, and these cells are actually harbored from that adult tissue, it doesn't have the character of forming cancer or tumor. So many people nowadays are utilizing adult stem cell for their research and many clinical trials. If you check into the FDA and also the American Clinical Trial Portal, you can see the many adult stem cells being investigated for the multiple therapeutical purposes in the modern disease. Okay, and now this is my main work. This is what my PhD. During my PhD, I have investigated the impact of the stem cell in modulating the immune system. Okay, when you say immune system, so we have hematopoietic stem cell. That hematopoietic stem cell can give rise to many mature immune cell. It can be T cell, B cell, NK cell, neutrophil. It can be also the eosinophil and basophil. So we just put it away the red cell and the platelets. So we will be more focused on the white cell. And why we need suppress the immune response. Okay, you have to know that not all immune response are favorable. Sometimes we have the autoimmune diseases whereby your immune system recognize your own body as a what you call as a foreigner, as an enemy. So it try to attack your body. So it will create the autoimmune disease. Okay, the autoimmune disease also can be the type 1 diabetes, and we have multiple sclerosis. Okay, and we also have other disease, autoimmune diseases. Okay, you so we can what you call say it continuously. So these are autoimmune diseases whereby the immune system recognize yourself as an enemy. And sometimes you also have to control the immune system in the case of transplantation. The transplantation, basically, the individual, the receiver will receive an organ from donor. However, the immune system of receiver, okay, you may can what you call fool the police, and you may can fool this the spy, but you cannot fool your immune system. Your immune system has high intellectual property. It can recognize any single foreign molecules, any single foreign cells in your body, and it will work so hard to eliminate that foreign cell. So that's why in the case of transplantation, when you don't have a fully compatible matching, when you have average matching, there is a higher chances that the immune system will reject the organ that been transplanted. And sometimes you have a chronic immunosuppression, which is imposed risk for infection. And how we can see the reason how we can evade how we can evade the immune system. So we can give the patient into chronic immunosuppression by steroid drugs, but these steroid drugs, they have their own side effect, and it's also imposed for a high infection, high risk for infection. And what will be that alternate way to what we call induce immunosuppression when needed? So that's how it's come mesenchymal stem cell. Mesenchymal stem cell is originally found in the bone marrow. Now you can find the mesenchymal stem cell any organs of your body, and these stem cells deliver a non-specific immunosuppression for a short time period. Okay, so you do not need to have immunosuppression for a longer time. You just need them for transiently. Okay, now the mesenchymal, as I mentioned earlier, all the stem cells, okay, need to be defined. So how we define mesenchymal stem cell? Mesenchymal stem cells should be positive for some markers like a CD105, CD90, and is also negative for the other markers like a CD45 and 34. They differentiate into a multiple cell type, and they also express the markers that indicate that they can differentiate into a multiple cell type. Okay, and so here what we have done is we investigated, okay, the effect of the mesenchymal stem cell on the lymphocyte. Okay, so my PhD is to control GVHD. GVHD means graft versus host diseases. So when we do a stem cell transplantation, what happens is the stem cell differentiate into T cells and B cell, and these T cell and B cell actually came from donor. Now the donor or the graft imposed an immune attack on the host cell. So how to control this GVHD? So this can be controlled by using mesenchymal stem cells. So what we have done is we co-culture the lymphocyte, okay, from the donor, from the graft, and with the mesenchymal stem cell we co-culture them. okay, after we co-culture them, and we measure the proliferation of the lymphocyte. So when we activate the T cell with the stimulator CD328, okay, the T cell proliferate very vigorously. But when we add more stem cell, the proliferation reduce. When the concentration or the number of stem cells that been added into the co-culture reduced, the inhibition also reduce. Okay, so it shows that the mesenchymal stem cell can suppress the proliferation or immune response of the T cell. So this immunosuppression, it is not confined to a particular type of T cell. Both CD4 cell and CD8 cell are equally being affected. Okay, this shows that the purple color one T cell with the stimulation, and the red color one is T cell with the stimulation with the mesenchymal stem cell. So without any suppression, see, the T cell can proliferate how many rounds? One round, second round, third round, fourth round, fifth round, sixth round. So they can proliferate 6 times. But when we add MSC, you see the number of the proliferation and also the volume of proliferation reduced in the presence of the MSC. And it is applicable for both CD4 cell and CD8 cell. Okay, so it shows that mesenchymal stem cell, regardless the type of the T cell, and also any T cell or B cell, NK cell, or neutrophils or monocyte, macrophage, DC, they able to suppress the immune response. So our group, the stem cell and immunity in UPM, we are pioneer in this research. We have conducted the impact of the MSC on that many type, an array of the immune cell, and we found that mechanism and how the MSC actually reduced the immune response by targeting the proliferation, activation, and effective function of the immune cell. So that's the first part of my my talk on immunomodulation is completed. Now we now we move to a second disease model. So we also work with the diabetes. As you know that diabetes can be classified into a two type. One is it all depends on two things, and the quality and the quantity of that insulin. When the quantity of the insulin is reduced, when the body could not produce sufficient amount of the insulin, you will have a type 1. When the quality of the insulin is reduced, whereby you have sufficient amount of the insulin, but the insulins cannot act on that target cell. Okay, they're not sensitive to the insulin receptor, or the quality of that quality we call type 2 diabetes. Okay, so the diabetes and one of the NCD, non-communicable diseases, which affect a huge population in this world, and so much of the research work and clinical trials are going on to cure diabetes. However, until now there is no cure to diabetes. We only can manage the diabetes. And the way to treat the diabetes is either you restore the pancreatic beta cell, or you can control the autoimmune attack by the autoimmune immune cell. But restoring the beta cell is quite a difficult process, whereby you need a cadaveric islet transplantation. But it is not an easy process, okay? And also it's a painful process, and sometimes you will never find a fully matched pancreas, okay? And also if it is a transplantation involved two different genetically individual people, then you require continuous immunosuppression. And sometimes, okay, beside having an option to the cadaveric islet transplantation, it's also possible that to produce a beta cell from stem cell. So many people have said that the pluripotent stem cell, for example, embryonic stem cell and iPS, can produce a beta cell, and also people have used the mesenchymal stem cell to reduce the inflammation. Remember, the type 1 diabetes and also sometimes type 2 diabetes is also due to the chronic immune response. So by using the mesenchymal stem cell, we can ameliorate the immune response and restore the pancreas function. So many people have, there are certain clinical trials being conducted, and it shows that MSC can be safe to be used. Either they come from same patients or from the different individual, and both MSC, either autologous and allogenic, are safe to the patient to be transplanted. And there is so many of clinical trials are actually being conducted, and the results are actually very varied. Okay, the results from these clinical trials show very mixed result. But to uh, but to uh, to conclude what we have found using the MSC in that diabetic treatment. So what they have shown is MSC doesn't differentiate into a beta cell. Okay, they never find that MSC can differentiate into beta cell, but what they have found is MSC reduce the inflammation in the pancreas. When the toxic environment such as inflammation is produced in the pancreas, the tissue resident stem cell within the pancreas, they can revive and they can give rise to beta cell. And second thing is MSC also can recruit and revive that tissue residence beta stem cell. Okay, and that's the one thing. And second thing also, people also using new way of treating the MSC. These are still under clinical trials, whereby the patient's T cell and B cell that is reactive towards the pancreatic beta cell are being educated by the MSC. So now they not being reactive towards this beta cell. And then my third one is a myocardial infarction and heart failure. And about 17 million people die every year. And it's one of the most common causes is ischemic heart disease, is imbalance of the oxygen supply and the demand. It kills, so when there is a myocardial infarction, it kills 25 of the cardiomyocytes, okay? So usually, so when you have the death of the cardiomyocyte, you have a two option. Okay, one is a quick fibrosis, other one's a regeneration. As you know that heart is post-mitotic organ, it means that it don't have so much of regenerative capacity. So in this case, what happens is the heart will take a quick what you call way to do a fibrosis. But fibrosis is not a cardiomyocyte. The fibrocyte cannot what you call contract. Okay, so in order to have a fully functioning or optimally functioning heart, you need to have a cardiomyocyte, not the fibroblast. Fibroblasts serve as a quick fix for the injury, but it cannot function as a cardiomyocyte. That's why in the case of the myocardial infarction, where you have a fibrosis, and most of the patients will have lowered cardiac function. And the moments we have a pharmacological therapy and also revascularizations. Okay. However, this cardiac regeneration can be achieved by using multiple type of the stem cell. Okay, we can use a scuttle myoblast or bone marrow stem cell. Okay, it can be any bone marrow stem cell. It could be endothelial cell. It could be a hematopoietic stem cell, or we can use a mesenchymal stem cell, or we can use the induced pluripotent stem cell, or we also can retrieve the tissue resident stem cell of the cardiac tissue, or it can be also embryonic stem cell. So people have used multiple type of the stem cell to regenerate the infarcted area. Okay. And this is one of our research we have conducted many years ago. So we receive, we have conducted this research work where for the last stage of the cardiac patient. So during the open heart surgery, we receive a small piece. Okay, it's very small piece of the cardiac tissue. From the cardiac tissue, we generate that the cardiac stem cell. So by day 10, we can achieve a sufficient number of the cardiac stem cells, and we characterize the cardiac stem cell using specific antibody like a STRO1 in CD29s, and also CD105. And we also look at that expression of the stem cell markers and also expression of the cardiac lineage marker. So we are successfully had generated the cardiac stem cell from that a small piece of tissue of the cardiac tissue that came from the patient. And then what we happen is, so then we produce them for within a month time, about 1 million of the stem cells, and then it will potentially to be used in the clinical trial. So it shows that for the cardiac defect and cardiac related degenerative diseases, okay, in order to avoid the fibrosis, okay, fibrosis is a quick fix for the myocardial infarction, but the fibrosis cannot restore the function of that cardiomyocyte. That's why we need to replenish that infarcted area with that, either by the cardiac stem cell or cardiac stem cell derived cardiomyocyte, or using any other stem cell. So this will restore the function of the cardiac cell. Okay. Now the last part of my talk will be how the mesenchymal stem cell being proposed to treat the modern disease which we have now, which is a COVID-19. As you know that SARS-CoV-2 is a novel coronavirus induced pneumonia, which named as a COVID-19. Okay. This virus, okay, has as an envelope virus with a spike protein, a single-strand DNA. The envelope has a spike glycoproteins bind to the cellular receptor angiotensin 1 converting enzyme 2 ACE2. Okay, you can see very clear. And most of that the vaccine nowadays are being produced against these spike proteins. So what happened is, usually a normal person, a person with that optimal immune response, the body can withstand the disease, and the body can heal, can produce an antivirus response in order to avoid that advanced stage of disease. Let's say patient with immune compromise, patient with a comorbidity, older patient, young patient, where they don't have a fully functioning immune system, it can cause them advanced level of pneumonia. It means it may cause lung failure. Okay, so in the case, what happened is the body start to produce multiple pro-inflammatory cytokines, and these cytokines produce a cytokine storm that cause a collapse of the lungs or the alveolar cell or the epithelial cell. So it means the lung will lose the function, so it is unable to exchange the gas that required for the body metabolism. So in this case, what happened is, can we use, because in this case you have to remember two things has happened. One is the extra-generated immune response, the body fully affected by the cytokine storm, the pro-inflammatory condition. The second one is the injury of the lungs tissue, epithelial tissue. Can we address these two by using the mesenchymal stem cell? So when the immune system is overactivated, production of a large number of inflammatory factors resulting in a severe cytokine storm, and it induces organ damage followed by acute respiratory distress syndrome, or ARDS, or acute cardiac injury, and also which may cause death. So preventing and reversing cytokine storm may be the key to save the patient with a severe COVID-19 pneumonia. Okay, and avoiding the cytokine storm may be a key for treatment for COVID-19, and MSC, owing to their powerful immunomodulatory or immunosuppressive ability, may be beneficial to this attenuating the cytokine storm. So this is the first clinical trial been conducted in Wuhan, China. So they have used mesenchymal stem cell. Okay, and before that, so what they have, because usually in that ARDS or the lung failure or the lung infection, what you will see is, you will see this kind of the ground glass opacity. So it shows that damage has been made, okay, as damage has been made in the patients. However, when they have used mesenchymal stem cells, see the damage is very severe. However, when mesenchymal stem cells to given to the patient, you could see that the damage made by the disease are actually disappeared. So it shows that either the MSC helped to restore the functions and also regeneration of the epithelial cell in the lungs, either through the process called restoration of the epithelial cell, or they also reduce the cytokine storm by inhibiting the immune response that due to COVID-19. Okay, so you can get, because I can go more detail, but I'm afraid that you may don't aware of the terminology. So I just summarize that the results which obtained from the first clinical trials in the China. And so there are many approaches of using MSC. Okay, one is cellular therapy, whereby we can use bone marrow, mesenchymal stem cell, umbilical cord stem cell, adipose tissue stem cell to treat that COVID-19. And sometimes we also can opt to the acellular therapy. It means MSC, because if you notice that the major therapeutical effect of the MSC actually come from the excretion, the soluble factor produced or secreted by the MSC. So what people have done is, people have cultured the MSC, people have their exosome. Exosome is a kind of a small organelles in the body that produced by the MSC. I mean, most of the cell produce the exosome. This exosome being excreted in that what you call culture media, and is being collected and given to the patient. Okay, so rather to use a cell to treat the patients, now people have moved to, they have taken that factor produced by the MSC to treat them. So this we call acellular. So we don't use cell, but we use the organelles, the cytokines, the growth factors produced by the particular stem cell to treat a particular disease. Okay. So why MSC lucrative for COVID-19? Because as you know, MSC they can home to the lung and safe. It is safe and it can mediate the lung reparative function in animal model of lung injury. And they also can be immunosuppressive. Okay, they can suppress the entire immune system and they secrete anti-inflammatory cytokine. They also show they produce antimicrobial substance towards the bacteria and viruses. Okay, and it's also inhibit the lung fibrosis. It regenerates the lung tissue, rescue the injured cell from the apoptosis, and enhance clearance of the alveolar fluid. And it's also produce extracellular vesicle that can be used as a what we call acellular therapy. However, there is a lack of evidence whether MSC can directly transform or transdifferentiate into epithelial cell is still lacking. So with that, I think I can end my talk. So what I have captured at the moment is that stem cell, okay, it has a huge prospect for treating modern diseases, chronic diseases, especially cardiac, diabetes, and immune modulation. And many research are still under the what we call clinical investigation. So what I show you is the prospect of stem cell. So when you listen to this, you may not surprise, you may what you call fully overwhelmed that stem cell can be done so much a thing. Yes, it can do so much thing. However, all this work should be properly subject to the clinical trials before it becomes a clinical application. So what I have explained to you, what I have mentioned to you just now, these are mostly are under the research or under the investigation, and where this has to undergo a proper clinical trials to become a clinical therapy. With that, I end my talk. Now I would like to have any questions from the, you may ask me the question from the floor.
Thank you, Dr. Rajesh Ramasamy, for the wonderful talk. Now we will have our Q&A session to answer participants' questions. I would like to invite the audience to ask questions to clear your doubts. Questions can be asked either by submitting it through Zoom chat box or unmuting yourself?
I think it's best to ask me directly. It's much interactive, you know. Be
Yeah.
a brave, bold. You guys young, right?
Thank you, Dr. Rajesh, for
Thank you.
supporting our students. Nice to see you after a long time. It's totally run by students. So yeah, very enthusiastic and very nice. Nice to see.
As I told you, there are a lot of aspects. People have used stem cell for neuron diseases, skin diseases, but I only touch those which I work with that. Okay, so don't think that the stem cell can be used for these 3 different kind of disease. It actually is huge. If you Google, the Google auntie will tell you a lot of story than mine. Any part, maybe you may have difficult to understand certain terminology, you don't understand certain explanation. If you have that, you always feel free to ask me. Or if you, or maybe in the future you want to do a master or PhD in a stem cell, always feel free to approach me. Who knows? You could be my successor.
There has been few questions asked through the registration form. So the first one would be stem cell. They asked if the stem cell got any bad effect to humans or not.
Bad effect in the sense that what kind of a stem cell? You can't simply blankly or blanketly say a stem cell. Stem cell, we have embryonic stem cell and also inducible pluripotent stem cell, adult stem cell. See, like when you utilizing the embryonic stem cell and also inducible pluripotent stem cell, these are very volatile stem cell. It means these 2 stem cells can become whatever they want. So when you're young, okay, they're very volatile. It means it can be whatever you want, but same time, because of this very volatile, it also have a plenty of what we call a system that control their differentiation. So many people, when these stem cells being transplanted into to measure the safety, they found that these stem cells can produce tumor. So that's why the usage of embryonic stem cell and also the inducible pluripotent stem cell in the clinical application and also in the research in a clinical trials are very less because safety issue. Although it has so much of the therapeutical prospect, but when safety is compromised, we can't use them for the research or even in that clinical application. Okay. And that's one of that, I would say that drawback of the stem cell, and especially those pluripotent stem cells. It's like, see, it's very simple. When you're young, when you're preschool, when you've been asked what you want to be, you'll say, I want to be a pilot, doctor, this and that, right? You're pluripotent. So let's say when you move slowly to your SPM, after going through all the trouble in life and you know where you are. So at that time, you know, most probably I'm not into a pilot. Maybe I want to be a biotechnician. So you choose, you become a multipotent stem cell. Now, from the pluripotent to become whatever you want, you slowly evolve into one single ambition and ambition or what you want to be. That's what happened to the stem cell as well.
Right.
Because when you're young, you think you can change the world, right? You can change, you can do whatever you want. When you're 5 years old, even you believe in unicorn and Godzilla and dragon, right? And this age, you know the reality.
Yeah.
You know there is no unicorn. You know there is no dragon, right? So you know you only have an iguana or lizard. Something like that. So that's the only drawback with the stem cell, those with the pluripotent stem cell. And remember, in Malaysia, not only the hematopoietic stem cell transplantation for leukemia patient is illegal. Okay, other stem cells are not legalized. Okay, because in private, I have no idea how they actually doing. But in Malaysia, if you wanted to have a stem cell therapy, that therapy should have completed the clinical trials and also FDA approval. So remember that. So many work in Malaysia on stem cell are still under the investigation, either in a laboratory investigation or preclinical or under the clinical trials.
Also, sir, there are several supplements of these stem cells nowadays. So, is it advisable to consume them?
What is that?
There are few supplements for stem cells.
See, you cannot consume stem cell. Whatever you consume, you'll be digested, right? What they meant is a stem cell enhancer, like a green algae, okay? See, the stem cell enhancer is not something new for us. In the Indian and Ayurvedic in Siddha medicines, we have the Gayakalpa herbs. That's a stem cell enhancer. See, in an Indian, and also in a Chinese medicine, they do have certain like a ginseng and certain herbs. They actually can enhance the number of stem cell in your body. So what these modern people are actually doing is they are preparing these components, a compound. It could be from any herbal formulation or mineral formulation. When you consume, it will increase the number of stem cells, but whether they are clinically proven, not sure, right? It's only just by being word of mouth. Maybe they have some little research, but it's not actual clinical trials or clinical investigation. But remember, our ancestors have done this. So in Ayurvedic and also in Siddha medicines, we call Kaya Kalpam. Kaya Kalpam means rejuvenation. Okay, and also certain herbs. I didn't include these in this talk. I do work on herbs and also stem cell, but I didn't include in this talk. But in our ancient system, we have our what we call specific nomenclature for the herbs that indicate which herb has this what we call rejuvenation ability. Same thing goes to Chinese medicine as well. Like an amla is one of the amla and turmerics are known to have what they call have regenerative potentials. And also maybe in the Indian like Karisalangani, which been used for that hair, it known to induce hair follicle stem cell proliferation. So yeah, can, but it not been fully scientifically proved.
And the next questions was, what are the obstacles that must be overcome before the potential uses of this stem cell in cell therapy will be realized?
Yeah, as I explained to you, you need a proper scientific investigation and clinical trials. Because clinical trial is not an easy process, okay? You require millions of money, right? Because you have to recruit the patient and patient being monitored, and it has been done with the different phases, phase 1, phase 2, phase 3, phase 4. And because of that, sometimes when we do clinical trials, whatever impact which you see in the animal is not being mimicked in the animal study. There are many what you call stem cell clinical trial being suspended because they don't see an expected impact on that human. So these are things, the lack of scientific study, the lack of clinical trials that hinder the stem cell therapy to stem cell treatment to reach the patients. And also it's not a very cheap treatment, because you cannot simply produce a stem cell in that lab. You require GMP lab. GMP lab itself require millions of dollars to have one, and the entire process. So maybe one vial of that stem cell, if let's say if it has been approved, it may cost from 10,000 to 30,000 of Malaysian ringgit.
There's a question in chat box. So the question is, stem cell use in surgery is rather aggressive in all fields of medicine or surgery. Do we have data to show their success rates, failures, and adverse outcomes?
I mean, what is stem cell use in surgery? Need to be very specific. I mean, you mean the organ transplantation or the or that the bone marrow transplantation? See, like okay, let's I explain to you the one of the most successful stem cell therapy. About many years ago, when people have leukemia, what you see in Tamil movie or in any Asian movie, most of that what you call in the climax, every hero or heroine will die of leukemia, right? because they say there is no treatment. Okay, that's the old story. But you see, the treatment for leukemia has become revisited after the stem cell therapy. So what happened is nowadays we can do the hematopoietic stem cell therapy, whereby we can remove all these cancerous stem cell and also cancerous cell from the bone marrow of the patient, and we introduce a new stem cell from patients, okay, which free from a cancer, and now the patient can have the donor's red blood cell and the donor's blood cell, which is free from the cancer. So the hematopoietic transplantation for leukemia shows is only treatment that can cure leukemia. So in that case, yes, it's work fine for that hematopoietic stem cell transplantation. But for others, not very sure, because for except for that hematopoietic stem cell transplantation, there is no other actual legalized stem cell therapy in Malaysia.
The next question is that, is there a situation known as chimera where a person is found that he or she has a different type of cell?
Chimera, when you have a transplantation, let's say in the case of the hematopoietic stem cell, yes, your chimera. It means your blood cell actually come from the donor, the second party. But the chimera doesn't mean that you and the donor are completely different. See, when we do transplantation, we do tissue matching, right? You cannot simply transplant any tissue into an individual. Okay, most of the time we will get a closest match from the family members, the sibling. The most of the time, the chances to have closest match come from siblings or that haploidentical from the parents. So the chimera which you're going to see is very little. The differences of genetics is very little. Yes, it is, but not like something you having human head and lion body like what you see in the mythos. That's different one.
I think that's all about the questions.
No idea. If you have any questions, you always can email me. I may write my email address here, so you can email me as well. I'm much happy to guide or help. So I have I have wait, let me do. I have written my email address to all of you. So I just want to ask you, do you really understand my talk? Is it too technical? Be honest. Sankita,
Yes, to be honest, I feel like you summarized everything so that we students can understand that in a better way, like it's not complicated.
yeah,
It is.
because this is usually for those for the knowledge audience, but I have no idea how much you have learned in your course. So some of you from first year, but doesn't matter. You see, if you listen to a talk and you understand everything, it's no fun. If you know everything, why you should be in university? You should be working, right?
Yes.
Your students, you meant to be not know everything. So so it's okay. Learn.
Correct.
Any questions?
I would like to know if Professor Ravi has anything to say about this. So I think that's all. So thank you, Dr. Rajesh Ramasamy, for answering the participants' questions. It was a great session. Before I pass the mic back to the MC, I would like to take this opportunity to thank the organizing committee for giving me this chance to serve as moderator. This is my first time in life I served as moderator. This experience helped me a lot to learn a lot. I enjoyed the role of being this moderator today. I sincerely want to thank our senior lecturer, Mr. Jivendran Chandrasekhar, who's also our ABSA advisor. Thank you, Mr. Jivendran Chandrasekhar, for our continued support. Ladies and gentlemen, once again, thank you everyone. MC Mr. Tan, over to you.
Thank you very much, Dr. Rajesh Ramasamy, for a wonderful and informative talk. And thank you very much, Ms. Anita Malapitian, for moderating the session. Now, I would like to invite the Dean of the Faculty of Applied Sciences, Senior Professor and Ravi Agent, to deliver his opening remarks. Now, I would like to invite Mr. Lim Yenta to deliver a word of thanks. Mr. Lim, please.
Hello everyone, I'm Lim Jenda. I'm proud to tell you that I'm a year 3 student of BSc Honours in the Biotechnology degree program of Ames University. The organizing committee has given me this wonderful opportunity. First of all, on behalf of the ABSA and Webinar Organizing Committee, I want to thank Associate Professor Dr. Rajesh Ramasamy for an enlightening and informative talk on stem cells in modern medicine. Dr. Rajesh Ramasamy, we are grateful for the time and effort you took to share your thoughts and experience with us. We appreciate it. Thank you to Senior Professor Manikam Ravichandran, Dean of Faculty of Applied Sciences, for showing your support to us by attending today's webinar. I want to thank Mr. Hugo, President of ABSA, and Ms. Vivian, Vice President of ABSA, for their help. I would also like to thank all the participants from various countries for joining us for this webinar. I believe that this webinar helped you to get some insights and learn something new about stem cells in modern medicine. I would like to take this opportunity to record my thanks to all members of the webinar organizing committee for their hard efforts. Working together as a team for this webinar was an engaging and enjoyable experience. Last but certainly not least, my special thanks to our ABSA advisor, Senior Lecturer Mr. Jivendran Chandrasekhar, for his valuable support. Ladies and gentlemen, once again, thank you. Mr. Tan, over to you.
Thank you, Mr. Lim. It was a great session today. Before ending the session, we would like to hear the feedback from all participants by sending the call on the screen right now. Report me, the evaluation survey will not take you more than 2 minutes. Your feedback will be greatly appreciated. You can either send the QR code or go to the link shown below. The link is posted in the chat box as well. After the completion of the survey, you will receive an instant participation certificate in a short while. And I will keep this slide running for 2 more minutes for you to send the QR code. We are looking forward to having you all join us again in the next section. Thank you again, everyone, and we wish you good health and safety.
Thank you.
Before
All the
we
best.
all leave, I would like to take a group virtual photo. So here we go. 3, 2, and
No, we just wait for 2 minutes for the participant to send the QR code.
all right.
I hope everyone has the chance to send the QR code, and I'm going to end the meeting. See you soon in our next webinar. Stay safe and have a wonderful day. Bye for now.
Thank you. All the best.
Thank you so much, Prof.